Good morning, everyone. We're going to let everybody into the presentation here, but we'll get started very shortly. Thank you for joining us this morning for a Rigaku webinar topic on new applications with Progeny 1064 Handheld Raman. Good morning, everyone. We're just letting a few more folks join us. We've got to admit one by one so that we don't have any crazy Zoom crashers, but we're very much eager to start this topic of today, new applications with Rigaku's Progeny 1064 nanometer Raman. Good morning, Joe. I love when you guys say hello to me like that. That makes me happy. I'm lonely. I miss other adults. Hi, Joe. Hi, Dermot. Hi, Jose. Very nice to see you all here. Thank you very much. Hi, Erwin. Good morning, everyone. What week of quarantine is it for you folks? Here in Massachusetts, it is week number 9 for us of quarantine, believe it or not. Maybe that's not the official number from the governor, but that's definitely the number from school, I can tell you, as a mother of 4 small children. So we thank you very much for taking some time out of your working home day, if that's where you are, if you're lucky enough to be in an office or a lab with other working adults from 6 feet away. Thank you for joining us from there too. We are here to talk with you today about our handheld Raman device, the Progeny 1064 nanometer. It's a great tool, and we want to talk about some new applications that we've been working on. So thank you again for joining us. We've got you all on mute. If you have any questions, please go ahead and ask those in the Q&A session. We'll be happy to answer them as we move forward through the presentation, or at the end, we might address them all like that. And certainly, again, let's get started. So, just to kind of give you a background, right? Bulk material analysis is the forte of Handheld Raman. If you're here to join us and learn a little bit more about new applications, wonderful. But there's no doubt that these Handheld Raman instruments, like Progeny, were created for bulk material analysis. And they're great for that. Well, because the portability of the solution allows you to bring it to the problem, right? It was always about removing the need to sample. They're built to be rugged. Dropping the Progeny is a secret that I'll keep for myself. But it has happened a couple of times, and these tools are made to be used in the fashion that they need to be, so they're very rugged, and specifically Progeny is anodized aluminum, and it can be completely washed down and sanitized, which is obviously very important nowadays. And then clearly the onboard analytics, that's really the key, right? Taking the spectroscopist that you had to have in the laboratory and putting inside the little box. That's what made these tools so available and used so readily in pharmaceutical applications. But historically, they just haven't given the curious scientists enough. From my perspective, there were so many times that I've been using handheld Ramans, traditional ones, that I just wanted to learn more, and I knew my customers wanted to learn more. So that's really the kind of the limitation of traditional handheld Raman, is that on-screen pass-fail analysis only tells us so much, right? And maybe we want to change scanning parameters. So the algorithms and scanning parameters on Progeny, that's fully in your control. And then graphical analysis on the tool allows you some awareness right at the time of testing that, I think a traditional spectrometer a traditional handheld spectrometer, I should say, doesn't allow you that, where Progeny really gives you that ability to look more closely at your data and control the experiment as you acquire that data. So that's really important to kind of making the bridge between the traditional application of Handheld Raman. I have a quick question for you. So we have some poll questions, if you don't mind taking a moment to answer them. Jen, if you please post the first poll question. Thank you very much. So if you have a Handheld Raman, I would love to understand what you use it for. We would all love to understand what you use it for, whether it's all the time, 24/7, incoming raw materials inspection, or if you are, doing mostly raw material ID, but maybe you do some other work with investigations and things like that. If you are doing packaging materials, I'd love to know if you're working on packaging materials. That's a really cool application. Anti-counterfeit, things like this, finished products. Wonderful. Okay, I see. So a lot of you do everything. About a fifth of you are working on everything. And then about a third of you are using your handheld Raman tool for 24/7 incoming raw material inspection. And that's, that's really the value proposition when we bring one of these kind of tools into your realm, is turn over those raw materials into your manufacturing line as quickly as possible. But there's so much more that you can do with a Raman, and I definitely want to talk to you about some of that today. So if I haven't made it clear, I really think that Progeny does a great job of bridging the gap as a versatile solution. Obviously, as a 1064-based nanometer-based instrument, the availability of information, because you're not dealing with fluorescence in your spectra, really allows you to just see more about your samples. And if you've ever seen a comparison between 785 nanometer spectra and 1064 nanometer spectra and the availability of information that is allowed with using a 1064, that's pretty evident. But also just the ability to do your analysis faster with huge lots of containers because you're dealing with less fluorescence. That's a really important part of it too. But then getting to the real scientific exploration. It's a fit for all purposes kind of solution, we like to say, because you can change all of your parameters. You can change your power, wavelength, things like this. But everything that you can do with a Progeny, you can do it right on board the instrument, and that's really the key too. Best practices for typical Raman analysis, we look at powders, we look at solids, gels, liquids, lots of different kinds of materials. But in general, Progeny has to be able to see the sample, right? We deal with translucent and transparent packaging and not opaque. We have to make sure that we don't have labels and stickers in the way. There's some basic practical information that no matter how much we want to apply the tool in different areas, we have to kind of keep these in mind. The other one that we really have to keep in mind is that, Raman in the handheld fashion like this, it's a bulk analysis form. So as much as we might want to use the tool as a handheld spectrometer, the ability of a benchtop tool will oftentimes be more capable than what a handheld tool could be. So that specifically applies to kind of minimum concentrations. And when you're talking about a handheld Raman, we always like to tell folks that 3 to 5% is really the minimum level of a analyte that you're going to want to see in concentration. It's got to be at that level to have a really robust method developed on the instrument. So trace and quantification applications, they're not ideal. Just, from me to you, very direct and honest perspective with a handheld Raman, trace analysis, it just pushes these kind of tools a little bit too far. But quantification, that's also something people often want to do with a tool like this. And I always say, let's think about the why behind the quant. Obviously, people want to understand how much material is in, or how much analyte is in a specific sample. But perhaps a set of fingerprints might be even a better solution to the real problem at hand. So let's talk about some different applications. I start with one of my favorites, because one of my favorite polymorphs, right? Chocolate. Chocolate is the polymorph that we all love because there's a specific chemical form, there's a specific polymorph form of chocolate that makes it shiny and smooth to the taste. When we're talking about the pharmaceutical industry, it really comes down to bioavailability, right? Different polymorphs will have different bioavailability and form. And so the ability of a tool like this to kind of differentiate those is pretty important. There are so many polymorphs out there in the world. There aren't many readily available to the common person, but one that you and I maybe have access to all the time comes in the form of a common antacid, Zantac, right? Ranitidine is a common polymorph. It comes in two different forms, form 1 and form 2, and they have some differences. And if we look at the bottom right here, you can see the Spectra overlay of those two forms of ranitidine. And you can see some small differences at the peaks, the small peaks at 680 and 1050, but then also the shape of the major two peaks there is very different. And Raman is great at telling the difference between these two. There's no doubt Raman can tell very easily the difference between these two materials just at a correlation coefficient threshold of 0.8. Very, very easy to tell the difference. When we look at kind of the realistic application of why you would want to tell the difference between these two forms, it comes down to, again, the bioavailability. Zantac uses form 2 because it is just much more bioavailable in that form than form 1 is. And so the ability of Progeny to be able to pick out if form 1 or form 2 is actually in the final product is really a key capability of a tool like this. It's a real world application that's not raw materials ID. It's looking much more specifically, and the ability to shift correlation coefficients and things like this in a polymorph application is really, really important, because sometimes that standard correlation coefficient cutoff of maybe 0.8 or 0.95, depending on where you have your methods set up, you might have to shift that to allow for kind of polymorph applications and identification. In this case, when you're looking at the two spectra below, you see very clearly that with Zantac and form 2, a really nice overlay of those two forms, where form 1 with Zantac on the right clearly does not show the overlay. So the correlation is 0.3 versus 0.8. It's a really distinct separation. I have another question for you. Thanks, Jen. So, do you at your company or somebody else at your company do any sort of quick confirmation testing at the manufacturing line or of your finished product or of intermediates, anything like that? Non-raw material identification, mixtures, things like this. I'm very, very curious. Okay. So it looks like about a quarter of us don't know for sure, and I appreciate that. About a third of you, about half of you don't do too much of it, but 10% or so do some of this work. And this is really interesting to me, because the more I look at how the pharmaceutical industry has shifted, you know, I've been working with folks like you over the past 10 plus years, more than that. And what I see, and what I'm sure you all see more and more, is the intermingling and the way that every company kind of has a relationship with another company, and business is definitely ebbing and flowing in the direction of fiscal maximization. I'm trying to be political here. But really it's about, contract partnerships, and the responsibilities in those contract partnerships, I think, are becoming more and more evident, that we have to do as much kind of future thinking about how we will approach our responsibility around the identity and verification of product material as they go from one place to the other. And I think this is a great application to kind of dig into. For me, I'm just always curious about it because I see folks kind of on the fly doing it, but I don't know how much part of the program it is. So it has a couple of different areas, right? When we're talking about process monitoring, first, the starting materials, doing quality control on those starting materials, it's very, very similar to just a general raw material identification application, but obviously not nearly as regulated, right? You also have the end of the line, so to speak, where finished product analysis can be performed, and that's really, really similar to RMID, and it's oftentimes a very revealing strength in terms of a 1064, honestly, based instrument, because you see so much information in your finished product that you might not see in your 785 base instrument. But then, screening, right? Screening is really, when people think about process monitoring, that is what comes to mind. And it's the ability of a tool like this. Can we get an idea if our process is moving forward? If chemistry is happening without taking samples, going to the GC or HPLC, doing that kind of level of work, can we get a quick snapshot of my process is moving along? And the answer is, it's easy to say yes, but we have to think about what it would really involve. And it's best to think about not, again, quantification, but a start, middle, end is always the best kind of way to think about it. And to think about, are there identifiable points within our chemistry, within the evolution of that process, that perhaps we can capture with a Raman spectrum? And if that's the case, and that's going to be specific to your application, then Progeny is a really excellent tool to be able to do that kind of snapshot, beginning, middle, end. And here's a really simple application. I should say, we have other examples, but unfortunately we can't always show everything we want to be able to show. But this is a simple reaction of methoxyphenol and epichlorohydrine. Got it right. In yellow and blue. And basically that's just a simple chemical reaction there that shows in the spectra below how readily the two starting chemistries and then the finished reaction chemistry, the reacted phenol, can be speciated with a handheld Raman instrument like Progeny. And then on the back end, finished product analysis. I know I've got some folks here that do this work ad nauseum. I'm saying hi to a certain Joe out there. But it really comes down to kind of full cycle product awareness, right? That's paramount. Nowadays, we have to understand the quality of our product from start to finish, even when pharmaceutical regulations aren't as clear as we would like them to be. And that's always going to be kind of the heart of our predicament in the pharma compliance realm, is that we have to be ahead of the game. We always have to do a little bit more than what we're told to do, because things will shift, and usually it's in the direction of increased awareness, increased quality control, and increased inspection. And specifically as a CMO or CRO, these responsibilities are super compounded. So looking at pre-pack and post-pack production and the intermediate products prior to tableting and encapsulation, that's super important. And then looking at the processed product prior to its packaging, maybe looking at blister packs and being able to tell the difference between an API laden product and something that's a placebo product. These are all applications that are real world applications that our customers are doing, and I'm always curious to kind of introduce you out there in the world to these ideas, because maybe there's real value in your organization to exploring some of these ideas with your handheld Progeny. And then preclinical trial confirmations, that's kind of what I mentioned there in terms of API and placebo product. Particle size monitoring is another area that folks have been very interested in working with a handheld Raman on, because specifically the FDA has recently come out with a couple of interesting notes about the dissolution rates. And the effectiveness of certain products based on their dissolution rates, and that certainly failures in treatment can occur, and how is that related back to particle size. And certainly, you know, we understand that the two are inherently tied together, but as a part of the incoming inspection process, it's important to also understand that maybe you could use Progeny to understand that component of your system as well. There are certain limitations, though, you know, very distinct limitations, and they aren't necessarily limitations of the tool, but limitations of the problem. And let me show you what I mean by that. This is a great example of, you know, when we can look at particle size differentiation with a handheld Raman. And you can see here, looking at, you know, two different particle sizes, you can see a difference in the particle sizes for citric acid. There's absolutely no doubt that a Raman like this can teach you that. However, what really is kind of shaken out in the system is that particle size is usually reported as a median, but the variance in that is just absolutely massive. And this chart here shows you, you know, if you look at VivaPure alone, the average particle size reported, it's you know, 160, but the variance is all the way from 50 to 400 microns. So the Progeny is going to reflect that variance, even though the label says that it should just be hovering around 160. So when we're talking about these, you know, large particle size distributions, it makes it impossible, for example, for us to tell the difference between things like lactose 100 and 200. It just is really, really difficult for a tool like a handheld Raman to do that kind of work. Same with like Avicel 102 versus 101. Specific examples can certainly be differentiated, but when you're looking at the model as a whole and you're looking at doing kind of a really robust validation of your system, you're not going to be able to validate robustly around certain particle size challenges. So it's something to think about. And then the vendor, I almost left off this note, in the work that we've done and the work that Suzanne Schreier, Dr. Suzanne Schreier, I should say, I apologize guys, Dr. Suzanne Schreier and Dr. Valerian Subata are on the telephone or on the line with us here to answer any of your questions. And they've done all of this work. This is not my work, I just share it because I talk faster. But Suzanne has discovered in her work that really the vendor source is much more of a variance to the Raman spectrum than the particle size. Cell culture media is another interesting application, specifically with 1064 nanometer based Raman like Progeny. There is a lot you can learn about cell culture media. You can differentiate different types of cell culture media more successfully in powder form. Again, we get into this complexity of concentrations being too low when we get into the liquid form, usually to get really robust validation between our method when we're talking liquid form. But powder form, it's really pretty easy to differentiate different types of common cell media. This is just an overlay of 4 of them here on the right side, from DMEM to, you know, typical hams and things like that. And again, this is all the information that's found when you don't have fluorescence as caused by a 785 nanometer laser source. When you're dealing with 1064, you don't have that red hump of fluorescence that's on the left there. And so you see all of this information that's hidden underneath that hump. And with cell culture media, it's an absolute imperative to see that information. And we see a lot, and we can differentiate much, but it's, again, the ability of Progeny not only to be able to be a 1064 nanometer based instrument, but also because it is flexible and versatile in its ability to adjust the correlation coefficient. That gives us the ability to build models right on the fly on the instrument, that I can differentiate these 4 different types of cell culture media that are very commonly received in these biopharma applications. And it's because, if you see on this correlation coefficient chart here, clearly we can differentiate certain media. The green boxes are where the correlation coefficient is clearly in the threshold. But when we look at correlation coefficient specifically for like F12 to DMEM or HAMS to DMEM, that's a place where we see that we have to adjust our correlation coefficient so it's higher than those numbers so that we don't falsely identify them for each other. And on Progeny, it's just a matter of going into the application and adjusting that correlation coefficient so that my model can quickly represent the information that I want to delineate. When you're talking about other kind of applications in the pharmaceutical realm, emulsifying agents, that's a commonly received type of subset of materials. So if you're looking at cetyl alcohol versus stearyl alcohol and cetostearyl alcohol, the distinguishable features between these materials are small, but they're certainly there. And again, when we look at the results for these materials, we can see that the prediction accuracy to be able to separate these materials was 100%. SIRS is another really hot topic area I wanted to address, because if you're working with Raman, or you Google Raman, maybe you come up with SIRS. Surface enhanced Raman spectroscopy is when folks are using very, very low concentrate applications. And specifically, it's found it's, a home in safety and security industries and drug applications as, drugs get stronger, as illicit compounds get more effective, the amount needed to use them or the amount needed to be able to be detected, it needs to be smaller and smaller and smaller. And this is where SIRS has kind of found its home. But in terms of pharmaceutical application, it also can really be thought about for like brand protection work and finished product work. We have seen some success using SIRS in handheld Raman. Now I want to kind of give an explanation of how it works for you folks if you're not familiar with SIRS. In the diagram here, it shows that this is like a colloidal application. In the case of like a gold nanoparticle colloid is being mixed with the analyte and then dropped onto a slide. And that's usually allowed to dry down, and so you get a coffee ring on the slide. And then Raman is used to analyze that coffee ring. Now, it's provided some awesome information for lots of folks, but unfortunately what the limitation around SIRS is that most of the active bands that we see are much lower, closer to like the 500 nanometer range. So spectrometers that work lower to that range, 532 and 785, they tend to actually work better because specifically as well, the colloids and the SIRS strips that have been developed are specifically for kind of lower wavelengths. Now we are very excited because we have some colleagues at the FDA that have shown with some awesome results using Progeny 1064. We're waiting on those results, and we'll be happy to share those with you as soon as we have them, of course. But we also are working with different companies on the possibility to develop 1064 nanometer based strips. We're also working on different applications that aren't exactly like SIRS, but the potential to maybe use thin layer chromatography plates and things like that. So there is a world of kind of lower level analysis that we're open to exploring with you as customers. And if you're doing that kind of work, please reach out to us because we want to understand more about the work that you do, but we're also working on it internally, and we're excited to share information as soon as we have it with you. This is a cool application that our head application scientist, Suzanne Schreier, brought up for us because we have a great relationship with a company that makes a peanut allergy medication, and their product is based on peanut flour, of course, and so they wanted to understand much more about different flours, and so we wanted to help them understand this. And Suzanne, being a really talented baker in addition to PhD chemometrician, had about 30 different types of flour in her home pantry. And so as an example of kind of correlation coefficient madness, she went ahead and pulled together this map for us. And it essentially highlights some selectivity kind of challenges and capacities when you're working with a handheld instrument like a Progeny or any other Raman. And really it highlights how there are often great spectra, but you might not be able to speciate different materials from each other, right? And in the case of the flowers, what we saw in the correlation coefficient map was just madness. So this is when you take the data from a Progeny and you pull it into a different kind of software package. You go into it with Omnic, you go into one of your PCA analysis packages, and you do a little bit further work. You look at the data a little differently, or you ask us to look at your data a little differently. We're happy to do that. This is something that we find to be a really awesome part of our work, is when folks want to do different applications. And this is a case where clearly when we look at PCA analysis, the nut flowers and the wheat flowers can be very easily differentiated. There's no doubt about it. And then you got the peanut flowers, which was the real key. Those are also based as legume flowers. They were also easy to differentiate. But when we start to look just specifically at the wheat flowers, we wanted to understand a little bit more. And what we came to understand was that we can't really differentiate wheat flowers from each other. So I'm going to click on that, go backwards on that again, just so you can understand. If we look at the right side there, that's a specific library built around just the wheat flowers that are in the grouping on the bottom right on the left. And when we add more data to that model, because this is the step, right? This is kind of validation of a model. When we add more data into that model, what we understand is that, unfortunately, the specificity kind of disappears and those clusters overlap. So wheat flowers, we can't differentiate. This might happen with some of your samples. And I go back to cell culture media. This sometimes does happen when we're talking about 50 different cell culture media, and sometimes the only difference between them is a very small amount of vitamins at the very lowest level. When we add more data to the model, sometimes this is exactly what happens. So we're here to support you in it, but there are real implications to the work. When we talk about, packaging analysis, this is something that, I know that more people should be doing, especially with the utility of Progeny and how easy it is to do this type of work. To be able to pull open a package and ID the liners or ID the different materials, this is definitely a quality control application that should be explored on your site. If you're using blister packs or any sort of liners in your packaging, the ability of Progeny to differentiate materials that you probably can't see the difference between with a 785 instrument, you should take a look. And the ability to use a hit quality index algorithm in addition to our wavelet algorithm or a mixture algorithm on Progeny. This is highlighted here too, because in the case of PVC and PVDC blend material, and then PVC, PE, PVDC blend material, forgive me for that. You can see here through this kind of overlay of spectra and then the correlation map on the right that Progeny can very easily distinguish these two materials. There is a caveat that it has to be done on like a darker side. The other side is, it doesn't have actually a difference in the material structure. It's only on one side. So when we look at a little bit more closely though, if you want to differentiate PVC, PVDC material from just the PVDC material, again, it's about making sure we have a robust method for the application. And if we have all of these materials at hand, it's very challenging to use a handheld Raman in a compliant fashion unless you validated those against all the materials. And so this is where we would have to understand, can my Progeny discriminate between a blend of PVC and PVDC and PVDC? Tongue tie. And when we look at it, what we understand is that the real difference between these two lays in a much lower range, that 200 to 500 wave number range. And so this is, a place where we just tell Progeny, look at that specific range, do a hit quality index algorithm to the spectra in that range, and the instrument can very quickly differentiate the two materials based on that simple model. And with a threshold of 0.9, it'll do it all day long. And so that's a great application to understand kind of another place where your Progeny can be used. And so I have one last question for you, just one. And it's a simple one, a yes or no. I kind of hope that everybody says yes. All right. So far, 2 of you have said yes. Do you have an application that you've worked on or that you're interested in working on that's anything that we've talked about here today or different things? Please, if you feel comfortable in sharing with us in the chat, please do so. If not, I'm going to leave you with some basic information around it. About 3/4 of you say, yes, absolutely. You have an interesting application. Can we talk? Can we talk? Please give me a call, give me an email. Let's talk about the work that you're doing. I would love to understand more of it and certainly support you in that effort. There's probably something about our tool that we understand can be done and more about your work that you can understand that can be done. So please, you know more about your work than we do, but we're here to help. So let's come together as a team. We're definitely of that mindset as a group. Let's think about what we have to consider though going into those applications. Can we get the samples to mimic the sample variability, or can we make them? I definitely know there are folks on the line here that have made their own product when they couldn't get it internally. So let's make sure that we are understanding the material names and the concentrations that we're looking for. And then let's collect some data. If you have a Progeny, let's talk about what we need to see in terms of the data to understand the application feasibility. Or if you don't have a Progeny and you have samples, send us the samples. It's a strange time right now, but we are still working. As long as they are not toxic samples, we are happy to take them and do some work with them. I have a couple of Progenies. My colleagues have Progenies at hand, and we are happy to look forward to doing some more specific application work with you. With that, I want to say thank you very much for joining us. You have been with me and with our group for a couple of these TOPIQ webinars over the last month and a half. And I'm very grateful for these opportunities to connect with you despite the global situation. And I hope that you find this time helpful and you go to our YouTube page, the Rigaku Analytical Devices YouTube channel for recordings of these webinars if you like. And please reach out if you have any more questions or information. Thank you very much and have a wonderful Thursday. Take care. Bye-bye.